Sir? Okay, sir. Sir, am I audible?
Yes, audible.
Okay. Good afternoon to all. My topic is isolation and characterization of biocontrol agents against potato cyst nematodes. Currently, I am doing my final year project at ICA CPRI. Introduction. Here we are going to see in general about potato cyst nematode. These potato cyst nematodes are generally present in roots and potato plant as well in the soil of the potato plants. These nematodes are multicellular, unsegmented and worm-like animals. They feed on the roots of the potatoes so that the potato plant, their nutrients get affected and the symptoms develop from the ground upwards. So I'm going to identify that some biocontrol agents which helps to reduce the PCN. So here I have taken two plant varieties. I have taken the rhizosphere sample because these plant varieties, Solanum cisembriofolium and Solanum mauritianum, these two are resistant against potato cyst nematodes as well as root knot nematode. These are the plants. These samples are collected from the same institute itself. By serial dilution technique, I have isolated the bacteria and fungus. Here, these are the media I have used for growing the bacteria and fungus. So for cyst inoculation, I have used B5 media. I have done surface sterilization for the cyst with distilled water for about 3 times. And I have placed the cyst in B5 media. The bacteria and fungus has been grown around the cyst. The bacteria and fungus has been observed, and then further it is inoculated with nutrient agar and potato dextrose agar to get pure culture. After that, different colonies of bacteria and fungus has been observed. From that, I have done quadrant streaking for bacteria and I have plated fungus in potato dextrose agar. Subculture technique. After inoculation, I have observed different colonies and I have kept in the BOD incubator for observance. I have also observed morphological identification of the bacteria and fungus by identifying good non-contaminated plates were taken for the morphology identification. It depends upon the size, shape, and pigmentation. And I have named as CPRSU B01 for bacteria and for fungus CPRSU F02 for fungus. And those pure cultures were made into slants for further preservation. Preparation of culture filtrates of rhizobacteria. So from the mother culture, I have taken a loop full of bacteria and I have transferred them into 25 ml nutrient broth and kept in orbital shaker for about 48 hours at 150 rpm and transfer them into centrifuge tubes to get the culture filtrate. That culture filtrate is that one I'm going to use for with assistant RKN to check the number of juveniles hatched. So I have made into centrifuge tubes, and the cell suspension and cell pellet was obtained, and from that cell pellet alone I have washed for 3 times and kept for 8000 rpm for about 20 minutes, and I have stored it for 4 degrees Celsius in refrigerator. These are the centrifugation steps. Hatching test I have taken finally I have obtained culture filtrate. From the culture filtrate, finally, I have obtained culture filtrate. That one I am going to use for hatching test. For one bacteria, I have kept a 3 replication by placing 5 cysts in one multi-well plate, and same as for RKN. These are the plates. And I have, for control, I have used water, nutrient bath, and PRD. As a result, I have got the number of juveniles in PCN and RKN are counted. To find the most effective bacteria, the number of juveniles in control is high, where in treatments are low. Those bacteria has been used for biocontrol against potato cyst nematodes as well as root knot nematode. Thank you. Thank you, sir.
Am I audible? Sir, am I audible? Okay, sir. So good afternoon, everyone. Here I am Lawrence to present my work under the topic Molecular Characterization of Fluoroquinolone Drug Resistance in Mycobacteria Clinical Isolates. I've completed my project in IRL TB Chest Hospital in Pondicherry. So we all know that TB is tuberculosis is a disease caused by a bacteria that are spread from person to person through that air. TB usually affects the lungs, but it can also affect other parts of the body, such as brain, kidney, and spine. In most cases, TB is treatable and curable. However, people with TB can die if they do not get any proper treatment. So sometimes drug-resistant TB occurs when bacteria becomes resistant to the drug used to the treatment TB. So this means that the drug can no longer be killed the TB bacteria. So my objective of my work is I've collected samples of fluoroquinolone resistance Mycobacterium tuberculosis clinical isolates from the patients with the possible multi-drug resistance TB from Pondicherry. And I've also given representative figures for the molecular characterizations to the FQ resistance of Mycobacterial tuberculosis using a sequence analysis of the drug targeting gene for fluoroquinones. And I've also compared a resistant pattern obtained by line probe assay and a midget culture followed by drug susceptibility test. So as my first assay, I've taken line probe assay. So line probe assay is nothing but it is a DNA strip method. In this, we can identify the Mycobacterium tuberculosis and it is associated genotypic susceptibility to rifampicin drug and isoniazid. So LPA is based on hybridization of PCR products from the patient's sample or a specimen to specific probe for wild type and mutant allele of genes involved in the drug resistance and have shown some higher specific sensitivity test. And LPA results can be based on a banding pattern and detected on a strip method in hybridization. So this is my first essay which I've done. So these are the samples which I've taken. Both pulmonary and extrapulmonary samples can be taken for a line probe assay. I've taken a sputum sample of about 5 ml and I've processed. So the process is directly from DNA extraction of pulmonary and pulmonary samples, then the multiplication of amplification method, then reverse hybridization in which membrane strips are coated with specific probes complementary to the amplified nucleic acid. After chemical denaturation, the single stranded amplicots bind to the probe. So they bind to this rifampicin and isoniazid drug, so that I can tell that patient is susceptible to these bacteria. Sorry, these drugs. So this is the DNA extraction method and amplification method, copies of DNA, and DNA hybridization method in which the drug goes and attaches to the specific probe. So based on the PCR line probe assay, identification of molecular Mycobacterium tuberculosis detects rifampicin and INH resistance in this case. So the CC, AC, and TUB, this is the detection of the TB. So this is my first process. And followed by, I've got 72 positive tubes, 72 positive samples, and I've taken my project to the next step called as midget culture. So in this midget culture is a system that detects whether or not a TB bacteria will grow in a presence of TB drug. So midget is nothing but a mycobacterial growth indicating tube which detects the mycobacteria. The midget mycobacterial growth indicating tube contains a 7 ml of tube in which I've done my project. This tube contains a OADZ enrichment, Panta antibiotic, and so and so. And I've taken the same pulmonary and extra pulmonary samples, and blood and urine samples have been avoided in this. These samples have been processed and they have been loaded into the midget machine, so that the midget tube must be continuously monitored either manually or by automated instruments until it turns positive. So this is the midget tube in which I've processed. These are the granules after grown inside this after 7 to 8 days. So it is a fluorescent compound is embedded in the silicon at the bottom of the tube. And the presence of oxygen is dissolved in the broth. How much ever the oxygen is obsessed by the bacteria, so much ever fluorescence is detected. So this is the Bactec instrument in which I handled my tubes in it. So I've loaded my specimen process tubes in this instrument and incubated at 37 degrees Celsius for 7 degrees Celsius for 7 to 8 days. This Bactec instruments helps to scan the midget tube for every 60 minutes and it helps to detect the fluorescence in the tube for every 60 minutes. And it also helps to determine the positive tubes in it. So these are the samples which have been collected for my project. The same sputum sample, approximately a 5 ml of sputum is collected in it. So the specimen is being processed by adding antibiotic supplement called as Panta, followed by, after dissolving of Panta, the mix is being added to the midget tubes, and 4 to 5 ml of sputum is being centrifuged, and the pellet solution is being mixed to the sterile PBS and added to the midget tubes. These tubes are being scanned and loaded into the instruments until it results positive. After 7 to 8 days, it turns positive. So after 7 to 8 days, this is the positive tubes in which the Mycobacterium tuberculosis has a growth in it. These are the granules, and this is the fluorescence end in the bottom. And this is a negative tube which can be discarded after 42 days. And I've also used a rapid kit for the identification, because it may tend to result as false negative result. So I've used this kit. This kit is used to tell whether the sample is contaminated, the tube is contaminated, or it has turned positive. So these positive tubes are being carried out to the next step called as DST. DST is nothing but it is done to find out which drug the TB bacteria and the persons are sensitive to. In this process, I've used second line drugs, that is kanamycin, amic acid, capromycin, levofloxacin, moxifloxacin 1, moxifloxacin 2, and linezolid. And I've used one growth control tube, and this is a sample tube. So in this, if the drug is against the mycobacterial, the growth will be inhibited, and the fluorescence will be suppressed in the drug tube. Meanwhile, if the isolate is resistant to the growth, there will be increased fluorescence in the tube and results positive. So the process of this DST is OADC, which is a growth enrichment supplement. OADC drug of known concentration, and the sample, a positive culture sample, is being added to these tubes, and these positive tubes plus one growth control is loaded to the AST set. So this is a picture of adding the OADC, that is the enrichment. So this is the workflow. So this is the loading of AST set. AST is nothing but the antimicrobial susceptibility test. Placing these tubes in appropriate carrier set, ensuring that they are placed in the correct sequence order, that is left to right. Midget tubes are being loaded to the Midget 960 instrument and monitored until it turns positive. So these are my results. So this is my second objective. That is, I've given a resistant pattern for first line drugs and second line drugs. So these these positive samples are being taken to the midget, and I've worked under these seven drugs, and so that I can tell that so this 1465 patient number is 1465. This patient is resistant to revofloxacin and moxifloxacin. So for his further treatment, I can ask him to go on with these both drugs. So I'm sure that this drug can be sensitive to the bacteria which is in his body. So I can suggest him this drug. So this is my project. So meanwhile, I've taken more than 50 samples and I have given a result. And that result explains about the resistant pattern obtained by the line probe assay, midget culture, and drug susceptibility test. So this is the line probe assay, that is the first line drug and the second line drug. This is midget and the DST. So thank you.
Thank you, Ms. Gladys. Now, let
Thank
me
you.
go with our online presenters. Online presenters, stay back with this meeting. Now, for offline presenters, I would like to welcome Ms. Kasturi for the method presentation.
Go there.
Morning, everyone. I'm Kasturi, first year for PhD under the guidance of Dr. R. Shankarpriya, PhD College of Arts and Science, Part 2. My topic for this presentation is in situ bioregulation of bacteria by using indigenous and alginate immobilized enterobacter. Coming to the introduction, heavy metals are considered as high atomic number and high molecular weight of wood, which is discharged by various industry into our environment. And when this heavy metal accumulates in our food chain and food web, it will affect the humans. So to get rid from this, the microorganisms which were dwelling in that particular environment was isolated and used for bioremediation. Coming to the bioremediation is a technique to remediate heavy metal in the environment by using microorganisms. Biosorption is an excellent bioremediation technique which uses metabolism dependent and metabolism independent process to remediate heavy metal contamination. Coming to the significance of foundry soil, I have collected a soil sample from foundry industry because foundry industry in India is the third. Significance of foundry soil. I have collected the soil sample from foundry industry because foundry industry in India, India is the third biggest industrial sector globally, followed by China and USA. In India, 50 million tons of metals are produced annually, which produce 25 million tons of waste that is accumulated in the soil. And in 2002, it was estimated that 94 million tons of foundry sand was deposited in our environment. The major heavy metals found in the foundry soil includes cadmium, lead, copper, nickel, zinc, iron, steel, aluminum, and brass, etc. Now, coming to the cadmium and its toxicity. Cadmium is a highly toxic heavy metal which is present in various anthropologists, foresight extract, foundry industry, phosphate minerals, electroplating, leather, and cement production, and pesticide fertilizer industry. These are some common microorganisms used for cadmium classification. Next. Coming to the objective, first I had isolated cadmium resistant microorganism and characterization the microorganism and optimize the environmental parameters to get the effective bioremediation of cadmium. Then several time analysis were done to identify the impact of cadmium on the cell. After that, the immobilized enterobacter there is immobilized cadmium resistant bacteria was developed by using algae, agarose, and cytosine for the effective removal of cadmium. After that, in situ bioremediation of cadmium was done by both clean and immobilized enterobacter. Next. Coming to the methodology, I have collected the soil sample from foundry industry and the concentration of cadmium in that soil sample was analyzed by atomic absorption spectroscopy. After that, by using serial dilution and plating method, that is cadmium amended plating method, I had isolated the cadmium resistant microorganism. And the minimum inhibitory concentration of the isolated organism, enterobacter variety, was identified. That is by using different concentration of cadmium amended plate. And after that, the characterization of enterobacter variety was done by using microscopic culture biophysical molecular analysis. Then the environmental parameters such as contact time, pH, temperature, dosage, concentration, and initial metallic concentration was optimized to get the effective metal removal. And the impact of cadmium on the cells inside the cell was observed in 7-10 analysis. And to enhance the cadmium removal efficiency, the carbohydrates such as algae, agarose, cytosol was immobilized with enterobacter variety. And that organism was identified for checked for the removal efficiency. And both the native and immobilized, algae immobilized enterobacter variety was inoculated into the soil in situ bioremediation was done, and the concentration of cadmium in the soil was analyzed. Next. Coming to the results. These are the areas collected the soil sample and it's cosmopolitan foundry and the concentration of the various heavy metal in that soil sample was analyzed in atomic absorption spectroscopy. Here, cadmium was 3.48 in that soil. By using serial dilution and metal amended cadmium metal amended media based method, 16 bacterial strains were isolated, which were cadmium resistant microorganisms. That among that, the highest MIC value, the organism which contained highest MIC value, that is 1300 ppm of cadmium was selected for all the studies. This is the antibiotic sensitivity pattern of the isolated organism. That is, once the bacteria have the resistance to anything, that bacteria also possesses resistance for antibiotic sensitivity, antibiotics too. That is called as co-selection property, because antibiotic sensitivity and antibiotic resistant and heavy metal resistant both is the same locus of the bacteria. Among used 21 antibiotics, this enterobacteriaceae shows 11 resistance zones for 11 antibiotics. Next. These are the antibiotics that are used. And this is the culture characteristic. That is, enterobacterial isolated organism was identified as enterobacteriaceae based on morphological and cultural molecular characteristics. It is a gram-negative rod bacteria which belongs to enterobacteriaceae family, and cultural characteristics in nutrient agar these organisms produce beige to white colored large circular and convex quality. In black agar they produce large smooth flat bodies with entire margin without any. Mesophia agar produce pink to red color uniform qualities. In violet red white agar it produces red color quality. In XLG agar it produces fair blue and yellow qualities. In standardized gel agar partially insulated pink to pink color qualities are observed. In EMP agar fair growth pink color qualities without sheen was observed. This is the biochemical characteristics results. This organism shown negative for indole MR and positive for BH citrate, and it is like produce acid but acid with gas production. It can ferment glucose, mannitol, sucrose, and not lactose. It produces negative result for urease, H2S, gelatin, oxidase catalase, and it produces positive result for nitrogen. And by using 16S RNA sequencing, I have determined the isolated organism as enterobacteriaceae. This is the phylogenetic tree of enterobacteriaceae. Coming to the environmental parameters such as contact time, pH, temperature, dosage concentration were optimized to get the effective bioremediation cadmium remediation efficiency. It shows that this organism can remove 50 ppm, 100% of cadmium at the 50 ppm initial material concentration, at the contact time of 42 hours, and at the pH of 6, and at the temperature of 20 degrees Celsius, and at the optimum dosage concentration of 1%. By this optimum concentration, enterobacteriaceae can remove 100% of nickel present in the medium. Next one. And this is the scanning electron microscopic image for cadmium untreated and cadmium treated microorganisms, in which cadmium treated microorganism, that is surface modification was occurred, and the shrinkage in surface area was occurred, which denotes that the cadmium was reacted to the microorganism. Next. In the transmission electron microscope, the cadmium untreated and cadmium treated cells were observed. Cadmium treated cell, that is the electron dense discrete region was observed, which is denoted as intracellular cadmium accumulation. That is the cadmium was intracellularly sequestered by the microorganism. And immobilized enterobacteriaceae, that is to enhance the efficiency of microorganisms. In carbohydrates such as alginate, cytosine was used to immobilize enterobacteriaceae. The first thing was alginate beads and atrosine. It is a phytosine. Under optimum condition, alginate beads with enterobacteriaceae can remove 100% of cadmium in a solution. Agarose remove only 86.87%, and phytosine can remove 91.08% of cadmium. Coming to the advantage of encapsulation process, that is, when the organism encapsulated, it release the microorganisms slowly into the soil, and it is non-toxic in nature. They are biodegradable and they are low cost, and it will resist the acidic environment. And in in situ bioremediation, I inoculate both the native microbacteriaceae and immobilized enterobacteriaceae in the soil, and concentration of cadmium in the soil was identified periodically. It shows that the immobilized enterobacal loyalty can bring 99.72% of cadmium in the soil, whereas native enterobacal loyalty can only remove 85.72% of it in that soil. This is the area where I use the executive bioremediation. This is the graph shows that the green colored line is the actually immobilized bacteria. In the beginning, 3.147 ppm of cadmium was present in that soil sample. After they took the treatment, after they took 0.0086 ppm of cadmium only present in the soil sample, the drastic decrease in the cadmium was observed. I conclude that biosorptions are effective by abiding technique for metal remediation due to the cost effective and environment-friendly nature. Hence, it could be concluded that an immobilized enterobacal loyalty was a potential candidate for in situ bioremediation of cadmium. I thought the future workers I have to do the bioinformatic analysis to identify the resistant genes in the enterobacal loyalty. These are some methods. Thank you.
Thank you, Ms. Kasturi, for your presentation. Next, I would like to welcome Ms. Priya for their presentation work.
Good afternoon to one and all present here. I am Priya, PhD Research Scholar of Department of Microbiology, PhD College of Arts and Science, Bangalore. I am pursuing my research under the guidance of Dr. Priya, and my research is entitled on in vitro and in vivo evaluation of wound healing potential of violation as a promising therapeutic agent against penicillin resistant Staphylococcus aureus. Next. So, as we know that acute and chronic wounds has become a serious health concern in the hospital-acquired environment, and from which microbial infection are the primary source of worsening the acute and chronic wounds. And in addition, antimicrobial resistance will also be another problem which insists researchers to find an effective alternative to heal the wounds, for which the microbial treatments was chosen, and microbial treatments in addition to the coloring property, it was also to exhibit various therapeutic applications which includes the antimicrobial effect. And among microbial treatments, violation was chosen because of their versatility in their antimicrobial properties to treat the wounds. Next, based on which my aim is to investigate the antimicrobial and in vitro healing efficacy of the violation against the clinically significant wound pathogen, especially MRSA. And based on which the objective was framed as to isolate the potential strain and to optimize the growth parameters for the maximum production of violation, to characterize the violation, to evaluate the antimicrobial activity of violation against clinically significant wound pathogens, and to determine the minimum inhibitory concentration and minimum bactericidal concentration against penicillin-resistant Staphylococcus aureus, and to perform the synergistic effect of violation with the commercially available antibiotic streptomycin, and to evaluate the in vitro toxicity as well as in vitro wound healing efficacy of the violation against in 3D mouse fibroblast lines, and to assess the in vivo toxicity and in vivo wound healing efficacy of the violation in crystal albinoid. Next. So based on which the methodology was framed to isolate and identify the violation producing strain, then it was optimized for the enhanced production of the violation. Then the violation was characterized, then antimicrobial activity was assessed, followed by which MIC, MPC and synergistic activity was performed, and then in vitro toxicity and wound healing assay was done, and in vivo toxicity and in vivo wound healing assay was done. Next. the strain which is said to be Cyanobacterium lividum that produce a predominant violation. And next, the optimization of growth parameters was done for the enhanced production of violation. Among various media tested, it showed the highest production in yeast malt extract agar. As it is an intracellular pigment, it failed to produce a pigment in the broth medium. That's why we are using an agar medium as a growth media. And yeast malt extract was found to be the optimized medium. Next. And pH 7 was found to be optimum for a violation production. Next. Temperature of 24 degrees Celsius was found to be optimum. Next. The maximum violation production was occurred at 48 hours, which is 2 days of incubation period. Next. And glucose was found to be the optimum carbon source for the violation production. Next. Various solvents were used to extract the violation from the organism, and among various solvents, it was found that acetone gives the maximum yield of violation. Next. So, under the optimized condition, violation was extracted. To summarize, the yeast malt extract agar was prepared and the culture was inoculated onto the surface of the medium. After incubation, the culture was scrapped from the surface of the agar and it was transferred into the acetone and to obtain a powder form of pigment, we have performed a lyophilization technique. Next. So this is the characterization of violation. For purification purpose, we have adapted column chromatography, among which 4 different fractions was found. And in that, the 4th fraction was found to be correlated with the standard violation, which was purchased from the Cayman, USA. And it was found to have an RF value of 0.76. Next. Human visible spectroscopy of the violation revealed that it has maximum absorption being at 575 nanometer. It was correlated with the standard violation. Next. FDR spectroscopy was done. The predominancy of amine and nitro group was found, which is structurally similar to the violation. Thus, it could be suggested that the extracted pigment could be of violation. Next. And in HPLC also, we have got a predominant peak retention time of 12, which revealed that it could be violation, which was correlated with the standard violation. Next. And in mass GCMS analysis, iododimethyl pyridinol group was found to be related with the violation compound, and thus it was predicted as violation. Next. So based on the 13C NMR shifts, the compound was structurally similar to the violation. Next. And 1H NMR also, there is a certain chemical shift which was found to have a micro group, which was found to be correlated with the violation structure. Next. In XRD analysis, there is a broad peak around the 20 degree angle, and it was found to have an amorphous state of the pigment was found to be in the amorphous state. Next. So antimicrobial activity of violation was performed against various clinically significant food pathogens, among which it showed the highest inhibition activity against the methicillin resistant Staphylococcus aureus. And thus the MIC and MPC was done for methicillin resistant Staphylococcus aureus. Next. Next. Minimum inhibitory concentration of violation was found to be 125 mg per ml. Next. And minimum bactericidal concentration of the violation was found to be 250 microgram per ml. Next. Next. And synergistic activity of violation was performed along with the streptomycin antibiotic, and it was found to have an indifferent effect, which suggests that the violation alone could be used as a potential antibiotic rather revealed that up to 2000 microgram per ml concentration of violation, there is no toxic effect in the normal mouse fibroblast lines. Next. In vitro wound healing activity of violation was done in 3D mouse fibroblast lines. It revealed that complete wound healing was occurred at 48 hours of contact time, which revealed that violation could be used in the formulation of wound healing agents. Next. So after that, we have prepared an ointment by using the violation. The ethical clearance was obtained from Cambridge College of Pharmacy. Then the grouping was done, and in which Mr. Angino rats was used as an animal model. That will be of 4 groups. One will be an untreated. Group 2 will be negative control, which contain only an ointment base. Third will be the positive control, which is an tramycin ointment that was commercially available to treat the wounds, and then the violation along with the ointment base was used. Next. So wound contraction studies revealed that at 21st day, violation exhibited 98% of wound healing rather than the commercially available ointment, which suggests that the violation could be an effective alternative to the already available synthetic drugs. And this is the picture showing the wound contraction studies. Next. So, in addition, histopathological examination of the wound was done, and we concluded there is a mucosal ulceration was found when it's still ulcerated at 21 day of treatment, and when compared with the tramycin, violation ointment showed normal epidermis at 21st day of contact time. Next. So based on which, I suggest that violation would be an effective alternative to the commercially available ointments with no side effects. These are all the important references which I referred for this work. Next. Thank you.
I request Ms. Priya to stay back. If anyone have any questions, you can ask.
It's a nice piece of work. We have a lot of data. Will you publish this?
Yes, sir. It is in process.
Central.
Yes, sir.
Which journal?
Bioorganic chemistry.
Biology. Okay. So go to the HPS. HPS. Actually, what is that? 3.162. What is that?
It is a derivative of violations, which is said to be a deoxy violation. It could be of deoxy violations.
Okay. The second one? 11.9 is up to 12.
That is violations.
Okay. It's outsourced.
Yes, sir.
Where?
Sir, at Karunya University.
Actually, the separation is not good. But anyway, outsourcing is not a problem. The thing is, the first peak, it's a dominant peak and it's a broad peak.
Yes, sir.
The peak should not have like this. Okay, you need to separate, you need to develop the method.
Okay, sir.
Otherwise, during the publication, you need to just copy this peak. Just avoid that peak. Otherwise, they will ask too much of questions. The next thing, this gas chromatograph, right?
Yes, sir.
Why you go for HPLC, then gas chromatography?
So gas chromatography to identify the compound which was present, and HPLC for checking purity of the extracted violations.
No, actually, I must you wait. HPLC and TCM, it's totally different.
Yes, sir.
No,
This is for structural elucidation purpose, right?
in
GCM.
TCM, then
Sir,
structure.
which means the violation, certain compounds will be present in violation. We have cross
Better go for emergency customers.
In GC itself, I have found the mass charge ratio, sir, which was found to be 3.4. That was also related to violations.
Violation of the did you check the standard?
Yes, sir. This one is standard and above is standard.
Okay. That is nice piece of work.
Thanks.
Thank you, Ms. Priya, for your presentation work. So, remaining participants can present in the afternoon session after the keynote speakers session overs. Okay, the registered participants will get their lunch tokens out of the registration spot. You can have your lunch and stay back here at 1:15. I repeat again, after your lunch, stay back in the MPN seminar hall at 1:15 p.m. 1:15 p.m. I repeat again, after your lunch overs, assemble in the MPN seminar hall at 1:30 a.m.